Antarianto, PhD, as the moderator, we will listen to an interesting subject which will open your participants' insight in biomedicine. Okay. Before we continue, I would like to share about term of condition for all of participants. Maybe admin can share screen. Okay, first, in our regular rules, participants should present a good manner during the event. Participants should leave their microphone in muted condition. Participants are allowed to ask questions through Zoom chat by mentioning name and former institution. Participants are required to fill in the attendance link twice to get the webinar certificate. The attendance link will be posted 15 minutes after the opening and 15 minutes before closing. And participants will receive webinar certificate only after filling both attendance link. So make sure you will fill the link twice in the first 15 minutes of our webinar, and the next link will be posted 15 minutes before closing. Okay. So, this webinar would not have been possible without the support of the DPASDP as the former institution. For that, let's listen to the remarks from the head of student program, Professor Asmarina, Magister of Science. Okay. Asmarina?
Okay.
You may start.
Okay, thank you very much. I'm so sorry that I'm late to join in this seminar because I don't forget, I don't see the link Zoom for this meeting, but I found that later. Okay, thank you. Good evening for all here, the participant, and also especially our distinguished guest, Professor Hanry Yu, to come to our study program. I'm Asmarina, acting head of the doctoral program in Biomedical Sciences, Faculty of Medicine, University of Indonesia. Yeah, for us, it is very nice to arrange this meeting, arrange this lecture, that we can gain so many knowledge from our visiting professor, such as Professor Hanry Yu. So we are very grateful for Professor Hanry Yu for all the willingness to become our visiting professor, and today will give us a lecture about your field and about your expertise for our staff and also for our students. And also here, I would like to express my appreciation also to Dr. Radiana for the initiation of this event. This event actually is supported by our University of Indonesia, to increase our academic reputation. So we would like to join and to make so many as possible collaboration with abroad institution to increase our capacity in research and also in academic reputation. So Professor Hanry Yu, once again, I would like to grateful for the willingness to become our visiting professor. In this program, we would like also not only give our student good knowledge in so many fields, in biomedical sciences, but also we expected that we can make collaboration in research, and also we expect also that you can, as a supervisor for our student for dissertation or thesis, And so maybe we expect also that Professor Hanry Yu can also as examiner for our student in the defense examination later. So in this evening, yeah, we very nice. We are very nice to meet you all here, and I think as I know, as far as I know, that Professor Hanry Yu has also collaboration with our staff, Professor Shana and also Dr. Radiana. So in this near future, maybe we can make a strong collaboration with our student as a doctoral student, yeah, in this field, yeah. So I think, yeah, I don't want to take too much your time and all here to hear or to gain the information, the knowledge from the Professor Hanry Yu. So I would like to say once more on behalf of this event, of this lecture for our distinguished guest, Professor Hanry Yu, and welcome. It is great to see you in this event. Yeah. So I hope this meeting or this event is useful for our student and concerning in the knowledge about the stem cell, and also here for the topic today. Yeah. So I give back to the MC to continue this meeting. So welcome for all, and especially for Hanry Yu. Thank you very much.
Thank you very much, Professor Asmaria, for the opening. We will continue to our main session, which is topic presentation by Professor Hanry Yu. Therefore, let me invite Dr. Dhewayani and Antarianto as our moderator, who will guide our discussion today. Good afternoon, Dr. Radiana.
I cannot hear you.
Yeah, your voice not so clear. Excuse me. So maybe you can echo.
Okay.
There is echo.
Perhaps it's okay if I take over from here, Dr. Oka.
Okay, yeah, you can take over. Yeah, Radiana.
Very much, Dr. Oka. So I will continue by reading the curriculum vitae of our professor, our visiting professor this afternoon, Professor Hanry Yu, PhD. He obtained his PhD in cell biology from Duke University, Durham, North Carolina, USA. And previously, his master and bachelor were also obtained from United States, from Washington University Medical Center for his MSc, and from Michigan State University for his PhD. Professor Hanry Yu obtained his professorship at National University of Singapore in the 1st January 2018. So I think you still have a long way in your professorship, Professor Hanry Yu. And he is also a director of NUHS, Research Core Facility, Confocal Microscopy Unit, and Flow Cytometry Laboratory at National University Health System. Among his very long list of positions, he is a visiting professor at Department of Biological Engineering, MIT, in 2014 for 3 years, and then also a graduate committee chair at MIT, a founding co-chairman, graduate program in bioengineering, National University of Singapore, as well as a very prestigious scholar, Human Frontier Science Program, long-term research fellowship at European Molecular Biology Laboratory, EMBL, Heidelberg, Germany. So with this long list of prestigious accomplishments, I think today we will learn more directly from you, Professor Hanry Yu. So the floor is yours.
Thank you, Dr. Radiana and Professor Asmarina, for your nice introduction. So what I will do today is to share with you a pre-recorded lecture, which was a bit better in timing. So it will be 40 minutes. After that, we will try to entertain some questions so we can discuss more with each other. So let me share the screen.
Good afternoon, ladies and gentlemen. Thank you for inviting me to give this talk.
Can you hear me?
I've been thinking about this topic of extracellular intelligence for the past many years.
It's a bit soft, huh?
It evolves from understanding how extracellular matrix will play a role in signaling cells and in controlling the cellular functions and tissue functions, especially in the liver as an organ. First, before I go into my talk, I would like to thank all the institutions listed here for supporting me faithfully over the past many years. Cell has been in the center of biomedical sciences since 1980s.
I'm sorry, I cannot see.
In the 1960s, after molecular biology
Can you hear me?
So prominent.
Can't you hear me,
in
Professor?
In the 1980s, I still remember when I first entered graduate school.
Excuse me, Professor Hanry Yu?
Cell biology lab.
Excuse
and
me?
the Journal of Cell Biology as a literature.
Yes.
Prof. Hanry Yu,
your slide is not so clear. We cannot see.
Yeah, your voice is not so clear.
So the voice is not loud.
No, I mean the slide, the PPT slide is not so clear. The explanation and the picture is not clear. So blur.
I see.
Yeah, we cannot.
Is it possible, Professor, if you directly present? I mean, the slide is still here, but then you directly present it.
Okay.
With your mic, yeah.
All right. So let me try to reshare the screen, and without the video, it might be a little easier. Let me share the screen.
Because I think the audio in the video is not so clear.
Yeah.
I see. All right. Let me try to turn on my PowerPoint.
Okay.
Okay. Instead of sharing screen like this.
Yeah, now it's clear. Okay.
Now it's clear, huh?
Yeah. Thank you very much.
Okay, so let me go back to share. Okay, now look at the slides. Let me try to explain it myself. So,
okay.
So basically, what happened is that for the past few decades, the biological functions tends to be focused on the cells. Okay, so in the 1960s, after the molecular biology becomes popular because of the DNA double helix, a lot of molecules has been playing a central role in the understanding of biological functions. And starting from 1980s or so, people start trying to understand cells in terms of its structures, in terms of its functions. And I still remember in late 80s, when I started studying cell biology, what happened is that the Journal of Cell Biology, as a specialist journal, was publishing articles on a quarterly basis. Every quarter there's one issue. And each issue has only a few articles. So in a way that you can understand that there has been a tremendous growth of cell biology in the past 30 or 40 years, right? So now people think about tissue functions, people think about cells. So everything is centered around cells. The illustration here, you can see that the inside is the cells, and outside is a white space. This is a typical textbook illustrations. So inside, we know a lot about molecular events, a lot of signal interacting with each other to respond to the environmental signals, such as growth factors, such as hormones, such as cytokines. And there are extracellular matrix outside, but our understanding of whatever is happening outside of cells tends to be very vague and simplified, okay? So in the past 10 years or so, as we wrote this book with my PhD advisor, Mike Sheets, we try to understand how cell is behaving in an environment which is both mechanical and chemical. And so as a result of that, our understanding is a little bit more advanced. For example, we start paying attention more to how cells interact with each other and then responds to the cellular, extracellular environment. But still, such understanding is a cell-centric. That means when you think about a tissue function, you think about the only thing matters is the cells. And whatever is outside of the cells tends to be a little bit vague, okay? So let me try to move this forward.
Was publishing in a quarterly manner with every, okay? So in other words, biomedical sciences and applications and basic studies
instead of
assume cell is the center of everything. We know quite a bit about it. We don't probably care as much as
what's outside of the cell in the future. So, because cell as individual identity is segregated.
Can you hear it?
At most, when we think about a population or collection.
Yeah, we can hear, but not so clear. Yeah.
Okay. Let me stop it.
It's not so clear, Prof.
Yeah.
Let me stop the sharing. Let me stop the sharing and let me open my slide. So it might be a little bit easier.
Yeah. Okay.
Prof, is it possible that you have not clicked the share sound in the Zoom setting from the audio in your computer?
I see. No, I have share sound, but it's very vague. But anyhow, let me try to turn on my PowerPoint slide.
Okay, Prof. No worries.
Okay. Okay. Let me share my screen. Can you see this clearly?
Yeah, we can see good.
Okay, all right.
Thank you.
Let me just give the talk by myself.
Okay.
All right. So basically, you know, this title slide was, I was talking about, I've been thinking about extracellular intelligence issue for a long time. So I'd like to thank all this institutional support for my research and my education for the past 20-some years. Okay. Oh, let me turn on my video. Okay. All right. Oops. All right, so I talked about the cell-centric approach to biology, right? Just before we stopped. I don't know how to turn on my video, but anyhow, it doesn't matter. Okay. All right, so I mentioned about the cell-centric approach to biology, but many people pay attention to the cells and not enough attention to the tissue environment outside of the cells, extracellular space, okay? In the past few years, there has been a tremendous growth and explosion of an area of biology called extracellular vesicles. The extracellular vesicles are vesicles which are secreted by the cells, and then they contain molecules, cytokines, even pathogens, or even nowadays drugs or genes to be delivered to the target cells quite far away from the donor cells. So there has been a tremendous interest in terms of the study or biogenesis, as well as the roles of extracellular vesicles. What do they do outside of the cellular space, in the extracellular space? And such studies has led to a lot of understandings of EV, extracellular vesicles, which can be exploited by pathogens, by, in this case, by parasites, to carry out certain stages of parasites' life cycles. So it's interesting to know that EV has opened up new areas of research to understand what is happening outside of the cells. And for a long time, people have been trying to study extracellular space by understanding the extracellular matrix, ECM. But such an understanding of ECM tends to treat the ECM as a passive matrix, as a supporting structures. Most of the studies are in the tissue, such as cartilage, because cartilage is an ECM-rich tissue. The cellular components is relatively small compared with the matrix. And so people are not thinking ECM, extracellular space, as a very active environment in terms of playing a functional or intelligent role in regulating cellular or tissue functions. In addition to the extracellular vesicle area, there is another area which has been growing recently, attracting a lot of attention, especially in the media in the West. That is the study of extrarenous RNA. Recently, there is a paper published in Nucleic Acid Research saying that there are certain extracellular ribosomes and tRNA which can shape the entire extracellular RNAome. That seems to provide some protein synthetic function outside of the cells, independent from the cells. This has generated a lot of interest because in the scientific community, there has been a large group of people believe life or the origin of species somehow started from the RNAs. RNA can function independent from DNA and proteins. So therefore, that has attracted a lot of attention. This data seems to suggest such RNA can be functioning outside of the cells, independent from the living cells. Then in the extracellular tissue compartments, there has been a lot of studies about the mechanics of it. So, for example, the few illustrations here try to indicate that exercise or shaking and rocking can cause body fluid, such as interstitial fluid, to flow outside of the cells in the tissue environment. And such shear stress or shear flow can impact the function of the cells. So in the original design of this kind of experiments, people are thinking that the mechanical environment outside of the cells are simply a non-intelligent kind of pressure or forces applying to impact the cells. Cells just passively or more actively responding to such an environment. But there is a pattern in such a shear stress flow outside of the cells. So to some extent, this kind of mechanical studies of the interaction with cells has begun to shed light into the more intelligent aspect of the extracellular space signals. This statement is a little bit difficult to comprehend, to understand. I will come back to this point later on as I try to illustrate in the rest of my talk. Okay? For example, the extracellular fluid space, most of the time, as I explained earlier, most of the time when we think about tissue, is cells and non-cell components such as extracellular matrix, such as hydrogel, such as, for example, hyaluronic acid and such, to resist the pressure and compression or shear pressures introduced to the cells to protect the cells from damage. As I explained earlier, such a gel or fluid-filled environment tends to be found mostly in the cartilage type of the ECM-rich cell poor tissues. But this is a study published in 2016 by a pathology friends whose name is NUHS in New York City. What they have found out is that in the gut, which is usually considered a cell-dense ECM poor tissue, and when they perform very detailed pathology studies, you look at the picture right, on the right here, in panel I. So they found there are fluid field space separating the cells and connecting to each other. So he sort of believed that it is an additional organ in the body instead of interstitial connective tissue, but it's the fluid which is connecting all the components of the cells together. Eventually, they might be connected to the lymphatic systems in the body. So such a understanding has begun to impose questions to the scientist. That is, when we think about the tissue, we are thinking about it's both the cells as well as the fluid-filled environment or gel-filled environment. They are equally important, and they are interacting with each other by some kind of both chemically and mechanically. So when these fluid compartments contract or expand, they will need to react by the cells, and the cell functions will affect how the fluid environment is going to behave. So instead of a passive environment on the active cell kind of view for the tissue function, people start thinking about a more equal role of the extracellular space in terms of what it does to regulate the tissue functions together with the cells surrounding it. Okay, so that's the beginning of such a probe into these questions. And if you look at the fluid field space in the body, in various organs, there has been such a fluid field space. For example, in the renal capsules or glomerular space in the kidney, there has been a lot of fluid field space where the blood is interacting with the cells and the urine is interacting with the cells. In the case of the pictures down here, for the microcirculation in the brain, the vessel contains very little space for fluid, and those fluid is impacting the endings of the microcirculations to interact with the cells in a very intelligent and autonomous manner. I have no time to talk about the vasculature here. But today I will focus a little bit more attention to the space in the liver. In the liver, there are vasculatures such as the sinusoid. From the portal region, right here in the center, the cursor disappeared. I should have got students to help me with computers. Anyhow.
Do you need assistance with the slide, Professor, or how can we help?
It's not the slide. It's my computer cursor. I want to point to my cursor to something. Anyhow, you just have to listen to me then. Okay. Okay. Here you see the. Okay. Did you see my cursor here?
Yes, we can see it, Professor.
Okay. All right. So, for example, in the portal regions, there's certain deoxygenated blood is flowing, combining with oxygenated blood in the vasculature called sinusoid. And then the exchange contact with the cells, then eventually leading into the central vein areas. Such a vasculature is somewhat leaky because of the sinusoidal endothelial cells has pores called fenestrations. So such a dynamic interplay between the fluids inside the compartment and the vasculature is actually quite equal in terms of the dynamics in the extracellular space, the fluid versus the cellular component itself. So in addition to that, what happened is today I'm going to talk a little bit more about biocanalakali space. So biocanalakali space, the cursor keeps on disappearing on me. Anyhow, as I said, my apologies for not able to show you the cursors. But the green area you see here is the bioductal space, eventually connecting to, in upstream to the smaller bio-containing space called biocanalakuli. And this space is the area which is providing all the contractions. Oh, yes. Okay. All right. So this BC is the bioalkaline alkali space. Bioalkaline alkali in the liver is the only source of contractions to expel the bile salt and the metabolites away from the liver into the gallbladder, eventually excreted out into the gut and out of the body. So in a way, for the many Asian people, they are talking about how to improve health by controlling the excretion of toxins outside of the body and things like that. But where is the original source of the force, which is squeezing all these metabolites outside of the body? It starts inside the liver in the bioalkaline alkali space. Because the bile duct as a tube, does not have a contractile machinery. They are a tube, more or less, containing the contents, but not squeezing them. So that's the reason why for the past number of years, my students and I, we have been interested in understanding how the contraction occurs in the bioalkaline alkali or BC space. So you look at the videos shown here, there are two pictures you see here. On the left-hand side, the picture is bioalkaline alkali looking in details. So they, over time, they contract, they expand, and they generate some kind of vesicles. And then eventually they will contract massively. So below that, below the video is a model of what happens. The model is like this. When the hepatocyte surrounding the BC is excreting bile salt into the BC, it will increase the osmotic pressure of the BC, and that will lead to the water to get into the BC because it goes in through aquaporin. And when the water goes in, they increase the size or volumes of the BC tremendously, as you see here in the middle of the cartoon. I really hate this because if I can use the pointers, I can really show you where things are. Yes. Okay, that's great. Thank you very much. So when the pressure continues to build up, what will happen is in some areas of the BC, especially the ending of the BC, there's no escape of the pressure. So therefore, what will happen is that the BC will try to resist the expansion, the growth, due to the influx of water, and the salt. So as a result of that, at a certain point, there's a breakage of the wall, which is lined out by ectomyosin bundles, and such a wall would be broken. So you start seeing those little blabs. And when the blabs usually the blabs can be recovering. But when the blabs stop recovering, then you will see a big vesicle spinning off in the rupture situations. And then at some point, because of associated with this kind of event, you will start seeing there's a massive contraction of the entire BC, causing the contents to be excreted out of the BC. And this kind of process continues throughout the BC. They eventually squeeze the contents into the bile duct, into the gallbladders. Okay, that's the mechanical models in a short period of time for us to understand what's happening. And this model, we see this kind of patterns in the pictures on the upper right-hand side. The green pictures is actually what we see in the mice, labeled or tagged with fluorescence actin. We can see this BC actually has several types of the BC. There's a BC. There is a long BC, there is a closed BC. The closed BC, you start seeing more vesicles spinning out compared with the long BC. Okay, right. All right. So let me move on to look at the BC itself more carefully. So when you image the BC, this BC in this particular case is in the in vitro culture situation. So we can study it more in depth. So in the phase contrast microscopes without any labeling or staining, you will see the BC because of the high salt content. It's very bright. So you can look at if you measure the areas of the BC in the pictures, you see that it's contracting and expanding in a periodic cycle with 30 to 40 minutes cycle in each cycle. Okay. And so if we label the actins in the BC and look at more carefully, what happened is that we start quantifying various parameters of the BC contractions. So let me go back to the original size. Okay, at the beginning of the BC, you see there is a lot of microvilli inside the BC. As the BC expands, those microvilli start disappearing. And as the area or volume increase, they start disappearing. And at a certain point, when the microvilli length, which is the black dotted lines here, reaches almost zero, what happens is the blue line. The blue line is actually the area of the BC. They stop growing. So, and also the red line is the actin intensity, which was labeled. So you start seeing the actin intensity start increasing. The bundle in the wall becomes thicker. So what I'm trying to say here is that from the quantification of this BC behavior, you start seeing that in each cycle, as the size of the BC increasing, they will stop growing when there is some kind of tension generated by the consumptions of the microvilli. And that tension triggers the flattening of the area increase. And at the same time, the cell will try to resist the changes by building a thicker wall of actin around the BC. So this leads to a more detailed model of what happens. And as you see that, you also notice that the blabbing, if we quantify the blabbing, so this is a more detailed analysis of what happens. If you quantify the blabbing, which is indicated by the black arrows here, the blabbing start increasing tremendously near the end of the second phase. That is before the massive contractions of the BC occurs. And when the massive contraction occurs, you start seeing the red arrow here. The red arrow is actually the BCV. This vesicles, which is spinning off from the BC, okay? And so this kind of quantification is very important in understanding the causal relationships between the pressure, the environmental signals, and the response from the cells. Now, one of the most important, let me go back to this picture, this three-phase diagram. You notice that among all these three phases of each cycle, there are many things can be studied, but one of the most intriguing things people want to know is what is the signal causing the contraction in phase 3, okay? Because if the contraction is the driving force for the livers to excrete all the metabolites and toxin out of the body, then I want to know what is the driving force signals causing the contraction to occur. And not only that, how does the contraction regulated? How is the contraction regulated is the most important questions people want to understand. So what happened is that people actually, we ourselves, as well as a few other groups in the world, have speculated for the past 20 years. It must be calcium signal which is regulating the massive contraction, because it's normal to hypothesize that's the case, but nobody could actually prove this contraction is occur because the calcium is triggering it. So what happened is the student actually Kapish tried to perform some studies on the hepatocytes using actin label as well as the calcium dye label. Try to see whether the calcium dynamics will coincide with the actin dynamics. One of the surprising things he found out is that most of the time when people who understand the calcium functions in biology, in cell biology, will know that calcium has the highest concentration in the endoplasmic reticulum and outside of the cells. But when people think about the calcium concentration outside of the cells, they are thinking it's an infinite pool of ocean. Okay, they don't think about calcium is localized in a specific compartment. To our surprise, what happened is that the concentration we noticed of the calcium in the BC is the highest in this kind of culture configuration. And we actually later proved that it's also in the same scenario in the animal situations as well. Now, it's very surprising to us that the calcium concentration is the highest in the BC, even higher than the intracellular endoplasmic reticulum, which is a large pool, concentrated pool of calcium inside the cells. So this kind of finding gave us the initial insight into hypothesizing maybe the calcium in the BC itself is the initial signal triggering the activation of the contractions. So this was a very big surprise because we always think about the cells as the initiator of everything, right? So what is BC? BC is a little space carved out by part of cell A and part of cell B, which is not an intelligent being by itself. But if the calcium concentrations in the BC is the driving force or initiators of the contraction itself, then the cell is a passive player in this particular case. So that is a fundamental change of the paradigm of, as we think about what is the role of regulating tissue function from extracellular space versus intracellular space. And in order to prove this, we basically have to study more carefully. So what happened is we look at the calcium dynamics by feeding the cells with dyes, and we look at the correlation between the BC contraction and the calcium intensities. It seems to be coinciding periodically between the calcium intensity and the BC areas or the volumes. Okay, so there's a correlation between the BC calcium dynamics and the contractions. Now, just because they correlate with each other doesn't mean that the calcium is triggering or causing the BC to contract. So to understand the causation, we cannot just look at the two phenomena in a parallel manner. So we have to more carefully use high-speed camera, high-speed microscopy to look at the dynamics of the calcium spike as well as the BC contraction spike. By looking at it more carefully, we notice there is a 6-second delay of the BC contraction after the calcium signal increasing in the BC, in the cells, and decreasing in the BC itself. That means from this data, it suggested the BC calcium leak into the cells and causing the cells to force the BC to contract. So obviously the contraction mechanism is still in the cells, but the signals seems to be coming from the leakage of the BC calcium back into the surrounding cells by about 6 seconds or so. So this was a very exciting moment in the discovery process because it fundamentally questions the intelligence of the extracellular compartment like BC. Now, to prove this further, we have done more careful and more detailed studies, such as asking the questions, if a cell has multiple BC surrounding it, and is there a traveling of the calcium signal from throughout the whole cells, and how is the contraction occur? And it seems to be, it's threshold dependent, because what happened is that only the high intensity calcium signals from the immediate vicinity, that particular BC will contract, okay? Now, we also try to investigate whether the endoplasmic reticulum ER is playing a role or not. So we use this a dye which collapses the ER, and they seems to have no effect. But we try to use a dye which is a general ionophore, which collapses the membrane potential. Then there is a collapse, dissociation of the calciums and the BC contractions. Now, let me pause here. So I'm still excited when I'm talking about this particular data, because it was really the moment which lead us to believe that there is a sufficient extracellular signals which is regulating the body's cellular functions in a rather intelligent manner. So that's the reason the paper's title, we call it Autonomous Regulations of the BC contraction by the calcium in the BC. Now, of course, people will ask the questions, what is the reason, what is the mechanism which is causing the BC calcium? There's a high concentration of calcium in the BC to leak into the cells. Is it because there is as the osmotic pressure continues to increase in the BC, obviously we see blebs, right? We also see the vesicles. That means there is a tension generated on the membrane itself. So if there's a tension, then mechanosensitive calcium channel is the prime candidate. So we test the localization of various mechanosensitive calcium channels in localizing to the BC membrane itself. Only piezo1 lights up in the pictures. So that leads to the model, which is illustrated in the panel E here. That is, when the salt keeps on secreting into the BC, when in the original state, when the ectomycin was more relaxed, they will start increasing the BC areas. And as more salt secreted in there, until you start seeing that the membrane is being stretched. And then after a certain threshold, the piezo1, which is a mechanosensitive calcium channel, is being pulled apart. And then there's a massive infusions of the BC calcium into the surrounding cells, or exodus of the BC calcium into the surrounding cells. And such entries of the massive amount of calcium leads to the local ectomycin contractions to occur. And how this is leading to the entire cell to contract, our data suggests that it's not contracting for the entire cell. It's really the local contractions surrounding the BC, which is called the driving force, leading to the excretion of the bile content into the gallbladder and eventually into the gut. Now, to prove this, it takes more time. So therefore, what happened is that we look at the certain modulators. In this particular case, the GSMTX4, which is a modulators of the piezo1. So by measuring the BCV is a representation of the pressures inside the vesicles, inside the BC. So when the BC pressure is high, vesicles frequency will be more. So what happened is that for the GSMTX4, which is inhibitors of the piezo1, then what will happen is you have a rapid buildup of the pressure in the BC. Then it will lead to more escape of the pressure through the BCV. So basically, they are unable to contract. When they are unable to contract, the BCV frequency will be increasing. So in a treatment for the some types of cholestatic environment, the bioregurgitation has been studied quite a bit. That is, when there's a blockade of the excretion of the bile salt out of the liver cells, people have been adding more bile salt, such as UDCA, to relieve the symptoms of the cholestasis, or especially obstructive cholestasis. So in such a scenario, people sort of always feel it's a little bit funny, because you have a cholestatic environment. That means you cannot remove the bile salt from the liver. But by adding more bile salt, you actually relieve the symptoms. So people don't understand why that is the case. And this study actually shed light into that. What is happening is that when you block by having a faster pressure build-up, that is blocking the efficient removal of the biosalt, you increase the chance of the biosalt escaping from the basolateral site in some form of the BCV, which is escaping from the blood-containing stream instead of coming from the bile stream. So by this kind of bioregurgitation, it seems to indicate that the study would lead to the results which clinically was observed. UDCA could relieve the cholestatic symptoms. Yeah, so for piezo-1 to activate the rescue of the BC contractility, we actually try to play with drugs, which is modulating the piezo activities. You notice that the drug which is inhibiting the BC contraction would actually have an effect in the I suddenly forgot about the role of UDA1. But anyhow, just bear in mind that this studies is modulating the D phenotypes. There are certain drugs which is affecting the BC contractions. And if you can use UDA1, which is a modulator. I think UDA1 is a constitutive activator of piezo-1, which is leading to the modulations of rescue of the drug effect. So to summarize, basically the understanding is that mechanosensitive calcium channels can regulate the BC contractions by colocalizing at the BC together with the high intensity of actin cables. And it's sensitive to the pressure, and it leads to the regulations of the massive contractions in the cells. So I'd like to thank the people who have done this work. In fact, my lab has been predominantly PhD students for many years until more recently. And more recently, there have been more postdocs than PhD students as Singapore start becoming tighter in training PhD students. The scholarship is very difficult to get for PhD students, but we do have very good postdocs. So I'd like to acknowledge the PhD students who have been doing great work. In fact, Kapish Gupta was trained in his master's and bachelor's degree from Indian Institute of Science of Technology, has been trained as a nanotechnologist, but he was retrained in his PhD program in Mechanobiology Institute and has published many good work. In his PhD time, he published one PNAS paper, one Nature Cell Biology paper, one Journal of Cell Biology, and the Journal of Hepatology paper. And now he's working as a postdoctoral fellow in UPenn, in my friend's Becky Wells labs in UPenn Medicine. And Qiushi Li, he was trained as a biologist from Tsinghua University. And he came here, published in a number of good papers, including a first author Nature Cell Biology paper. But he has a heart for entrepreneurship in his mind. So when he went back to China after graduation, he started a company called Cure Genetics, together with students from Zhang Feng's labs in Broad Institute in in MIT, and they are growing this company quite well in China. Yingchuan has been a Malaysian student studying in Singapore, and he did quite well in his PhD studies. He went away for a few years before he came back to my labs to work further in developing the mouse studies for the in terms of knockout mice and a few other mice studies. And Gauri Shenda is a wonderful postdoc from Indian Institute of Science. My lab is quite diverse. This is a small group of people working on the particular project I'm talking about. I would like to also thank my collaborators, Virgil Visnov. He is a physicist by training, and he has been picking up many of the alkali studies, and we collaborate quite a bit. Alvin Chen, my friend in NBI and Department of Biological Science, has been a cell signaling person trained in New Zealand, and we have been working with each other for a number of years. And I'd like to thank the funding support from various agencies. As you heard me, that I have multiple affiliations. It's not because I really want to be affiliated with many institutions. I don't have a choice because they provide funding support for my work, and also provide infrastructures and expertise that I can collaborate with. I used to run my lab independently in one site. So I have about 40 people in my lab for about 20 years. But I start realizing no matter how big my lab is, I can never be expert in everything. So instead of running a big lab, I start running multiple small labs. Now my lab has about 12 to 15 people, and split into 4 or 5 different sites. So we integrate into different infrastructures and expertise of different institutions. And that becomes much more powerful than myself running a huge lab myself. We can do many more things than we can do it before. Okay, so let me see, what's my time? Almost time is up. Okay, I have about 20 minutes to ask and answer questions. Okay, all right. Thank you for, there's some more slide you probably don't want to go through.
Thank you very much, Prof. Hanry Yu. This is a very revelating presentation, Prof. Hanry Yu, since all of us who studied biomedicine are very much a cell-centric person, cell-centric scientists or researchers, because we tend to think that it's all started in the cells, and the cells communicate with each other, and the environment, the microenvironment, extracellular matrix are only passive bystanders. So with your presentation today, it shows us that a tiny ion such as calcium can have a big impact on the contraction of a very vital structure such as bile canaliculi, which can prevent the cause of jaundice or biliary stasis. So it's really revelating, Professor Hanry Yu. So I would like to invite first our students to pose questions. I think I already saw in the chat someone typing a very long question. Okay, and this candidate is also the one that is very keen to visit your lab sometime next year, perhaps, Professor Hanry Yu. And she is also willing that if you would willing to invite you as her external reviewer for proposal examination, if you please. Okay, so I would like to invite Wahyunia. You have further questions?
Yes, Doc. I have already write my question at chat.
I see. Let me read it. Okay.
I think you can directly question
Okay,
directly. Yes.
Thank you, Dr.
Directly ask to the Professor Yu. Yeah.
Thank you, Prof. Hanry Yu. My question from about this guest lecture is about the phase 3 BC contraction that you said the calcium is accumulate in phase 3. The calcium itself, is it from intracellular calcium or from extracellular signal? That's my first question. And the second question, I just read about your paper about a vascular liver chip for sensitive detection of nutraceutical metabolites from human pluripotent stem cell derivatives. And we, with Dr. Nana also, we developed the iPSC-based liver organoid. So we want to know about your opinion. What is the advantages of liver on a chip, and in comparison with liver organoid, where all the cells were co-cultured directly? How do you consider the advantages between those two methods?
Okay, so you have a third questions and fourth questions, and later on we'll address them separately.
Maybe one by one, Prof.
Yeah, that's easier for me. Okay, firstly, you have very insightful questions. Even though we prove in this paper, the source of the trigger for the contraction is the BC contraction, which is the extracellular space. But if you ask where the origin of the signal comes from before they are accumulated over the threshold to trigger the contraction, whether they are from the extracellular pool or they are from the intracellular space, my gut feeling is that probably is accumulated by as a part of the secretion of the bile salt into the BC space from the cells. So to some extent, it's still cell-centric, but the trigger, at least the intelligent trigger for the contraction, is from the BC itself. You understand what I'm talking about?
Yes.
Right? So it's if you ask further questions, then it's probably still coming from the cells. Even though I disagree with my students, he actually believes that it's coming from the extracellular space. It's a lot of fun to interact with students, and as we explore the frontiers.
Prof. Hanry Yu, has there been measurement of the calcium ions between extra and intracellular in your system, whether it's coming from the hepatocytes or the extracellular matrix area? Did you measure them accordingly or not?
No, the reason why I believe it doesn't come from the extracellular space, the culture configuration, is because for the animal study, it's difficult to determine where is the source. But from the cell culture studies, in the calcium, the contraction, it doesn't change in the freshly changed media, which doesn't contain high concentration of calcium. In the calcium-free buffer, it's still okay. So the contraction still occurs. And it's regulated probably through the periodic secretion, continuous secretion of the bile salt along with the calcium from the cell itself. But that question is a little bit difficult. The reason why my student actually challenged me on that is because usually the excretion of calcium from cells probably have to be through STIM1 mediated process by fusing the ER membrane with but usually that process is uptake of extracellular calcium into the ER rather than the excretion of the ER calcium into the cells. So we don't know, to be honest. We haven't done careful studies of that. After Kapish left, I have too many exciting projects in the lab. So many students choose easier project than this. So currently no one is working on this project further. Yeah, so anyhow. So that's the first question. Second question is regarding you mentioned about the liver chips and in comparison with liver organoid, especially with cells from iPSC derived organoid, right?
Yes.
So the advantage, how should I say it? There's no conflict between the two. In fact, what happened is that recently we have been developing a NASH chip, non-alcoholic steatohepatitis chip, using by building a chip which can grow organoid and differentiate on the chip. And so we haven't published that data yet. And we demonstrated the in situ on chip differentiation of cells can be better controlled to have higher level functions, and then if you do it outside of the chip. The primary reason for that is because chip has a much better controlled soluble environment, but it's much harder to use compared with just the organoid in the ultra low attachment plate. So it's a very good question, but it's complex answers to the question. Yeah, I don't think there's a contradiction between the two. Okay.
Okay, Prof, thank you. Maybe my third question about the chip itself. I saw that it is designed with two parts for the upper cell and the lower side. And I want to know which signaling pathway that can be observed in high fidelity using organ on a chip if we are going to differentiate iPSC into hepatocyte. Can we investigate the signaling pathway, Prof?
Right, so there are two different chips. The chip you talked about, the vascular chip, is a relatively simple design because the vascular chip has the endothelial cells, vascular endothelial cells sitting in two-dimensional space versus the liver cells, which is sitting in a slightly different configurations. The iPS-induced cells, if you want to have control of the in situ differentiation, the chip design has to be different. So therefore, you can properly control the soluble environment with the delivery of the right growth factors and cytokines and such in the right time. So depending on the needs, the chip design has to be different. Yeah.
Okay, Prof, maybe my fourth question. Is this okay, Dr. Nana?
Yes, please.
As I mentioned before,
there's still no further questions in the chat room yet.
As I mentioned before, our research right now, we develop iPSC-based liver organoid. I wonder the size of the cells that can use in liver on chip, Prof. Can we input the liver organoid, which is size 500 micrometer and to 1 centimeter to liver on a chip?
Okay. For anything which is larger than 200 micrometers in diameter, the mass transfer between the side is going to be, so that means the center of the line will help a little bit in the perfusion chip because you do perfuse the liquid through the organoid. It probably can increase to about 250, maybe maximum 300 micron. If you want to grow a larger chip and the larger organoids, like 500 micron to 1 millimeter diameter or dimensions, then what will happen is that the center will become necrotic core. Necrotic core is good in terms of cancer research for multicellular cancer spheroid. But for organoid research, the only studies people have done is use to culture brain organoid. Brain organoid, the reason is not because they want to generate the central core as a necrotic core just to model the cancer organoid. But because when the cell dies in the center, you generate a gradient of environment from the outside to the inside. So that gradient of environment seems to be very important in defining what is the region of the brain that you're trying to model. Okay, so people use hippocampal neurons to grow them in the organoid chip in a larger scale. So either in the chip or in the well, they usually grow much larger brain organoid. And for the liver, it's a little bit, the only people who are interested in the liver organoid with larger organoid is people who want to study the zonation. You know, in the liver, there is a zone 1, zone 2, zone 3 from the portal region to the central vein region, right? So the oxygenated blood get consumed, and there's a gradient of matrix from zone 1, which is rich in certain matrix, to zone 3, which is poor in certain matrix. And as a result of that, the zone 1 has more progenitor cells, whereas zone 3 has more mature hepatocytes. So those zonation theory has a significant impact in terms of understanding liver cancer's treatment, because if liver cancer occurs in zone 1, you don't want to kill it. If you stimulate the immune response, you activate the zone 1 progenitor cells, you have more cancer stem cell to proliferate, which is dangerous. Whereas if zone 3 is where the cancer is, the treatment of zone 3 can be more bolder, right? Because you're not going to activate much of the signals in zone 1. So those studies can be properly modeled by a larger organoid. So one thing to remember is that model is a model is a model. A model is not everything. You are not building a whole liver outside of the body. You are just modeling some of the interesting features of the in vivo physiology or pathology. In fact, there's another student in my lab who is a mechanical engineer turned into a biologist, and she has been modeling hepatocellular ballooning in vitro, and the Gauri, the postdoc I just mentioned, she has been modeling bioduct development. So it's a very interesting exercise to model in vivo physiologies or development process or the pathologies of the disease.
Okay, thank you.
Okay. Thank you, Prof. Hanry Yu.
Welcome.
Okay. Nia, I think we can let other students to have the opportunity to ask questions. Or if there's still no further questions from the students, I would like to invite Professor Shana, Professor Rina. Are you still here? Prof. Rina, Dr. Aniretno.
Yeah, of course.
Dr. Novi, yeah, if you have a burning question, you can ask first while waiting for the students to come forward with their questions. Are you still monitoring, Prof. Shana? Prof. Shana have met Prof. Hanry Yu directly a couple of years ago.
Yes, thank you. How are you?
Good. It's
Yeah, I'm sorry I was late because I have trouble looking for the link. It happens often to me. Usually if I want to enter the Zoom, I have to look for the link first and I forgot where it was. But yeah, because there is trouble at the beginning, so I can see all of your presentations. Thank you. It's very interesting, but I think it's new for me that the bile canaliculi can contract. Yeah.
Yeah.
That's a very interesting presentation. Thank you.
Thank you very much. It was nice to see you again here. And I understand that nowadays there are too many Zoom meetings, so the links are flying everywhere.
Yeah, it's very disturbing because the link, there is sometimes in a WhatsApp group or sometimes in the email, and I forgot where it was.
I understand. Yeah.
Okay.
Today's talk is a little bit more biological. I hope that the next week's discussion will be more medical oriented.
Prof. Hanry Yu, so Professor Shana have a PhD student, right, Prof, who will completed her dissertation hopefully this semester on liver organoids, and the size that we have mentioned before is actually the organoid that we have designed in the lab. And as you notice that to characterize this organoid needs further advanced imaging, and perhaps that is something that we also could collaborate further on with Wahyunia's project.
Right.
It's also under the supervision of Prof. Shana.
Yeah, that would be great. We do have in Mechanobiology Institute and also in the core facility that I'm directing, we do have a lot of infrastructures as you have visited before.
Thank you,
Yeah,
Prof.
maybe we will need your suggestions later.
Oh, sure, no problem.
Thank you.
My pleasure.
Okay.
Yeah,
so,
I would like
Prof.
also
Rina?
I would like also to comment for your it's very interesting lecture, yeah, because as Professor Sana said that it is new also for me to know about the extracellular matrix, yeah, because I teach cell biology only the cell, yeah, inside of the cell. So, and actually there is also extracellular matrix that can also play a role in the cell function and also regulate some ion channel and so on. So it is amazing for me to know, and with your so nice picture, yeah, that has the capture by I think sophisticated camera, yeah, to evaluate this moving and this motility of the extracellular matrix. So I read also your publication that there is a small extracellular vesicle and large extracellular vesicle. Oh, why it should be divided into two kind of the extracellular vesicle. Is there any special function for this small and the large for the cell physiological function, or what's the important characteristic of this extracellular vesicle?
Right. So they are not the typical extracellular vesicles people are talking about.
Mm-hmm.
They are the results of the pressure buildup which pinching off from the cells, from the biocanalicular space. Mm-hmm. So what happens is that the smaller ones are the blebs. Mm-hmm. So as the membrane as the actin cable breaks, so the pressure will push the membrane into the cells. Mm-hmm. So what will happen is that, but membranes don't like to be alone without the actin cytoskeleton underneath attaching to it. So what will happen is they would quickly repair by building a wall around the membrane to squeeze the membrane back into the BC space. Mm-hmm. So it's a repair mechanism. So because it hasn't reached very big, then it appears as a very small blebs. But if the blebs becomes too big, they cannot repair, then it becomes a vesicle and just pinching it off. So the bigger vesicle is the vesicle. Smaller vesicle is just blebs.
That's okay.
Right. So it's not a typical ECV or EV that I talked about at the beginning of my lecture. Many people studying extracellular vesicle, they're much smaller in size. They contain molecules, peptides, even RNAs, and to signal far apart cells.
Okay. Thank you.
Thank you.
For your explanation.
Okay, thank you so much, Prof. Unfortunately, due to the time constriction, it's already 5 p.m. It's really interesting talking to you and discussing about about advanced imaging and liver on a chip, liver organoids, as well as extracellular intelligence. It's a new revelation. Thank you very much, Prof. So I will now leave to the master ceremony. Dr. Oka, are you still monitoring, or do you still have difficulty with your mic?
Hello? Can you hear me?
Okay, perfect.
Yeah, we can hear.
Okay, now I will leave the closing to you, Dr. Oka.
Okay, thank you very much, Dr. Radiana. Thank you, Professor Hanry Yu, for the presentation. We're now having a very good understanding about the extracellular matrix and how it works and how it interact with the cell.
Yeah, the voice is also not so clear.
Okay. Then okay, can we just give to Professor Rina for closing statement, Prof? Conclusion of this visiting professor lecture. Thank you, Prof.
Yeah. Of course, we thank you so very much for Professor Hanry Yu that has delivered the knowledge, the new interesting lecture today for us, for not only for the student, but for me also, especially because sophisticated research, I mean, with the nice picture and nice, of course, equipment, I think. So once again, thank you very much. And we hope that we can continue to make the collaboration such as the research collaboration or the joint supervision for our student here, Professor Hanry Yu. And thank you very much for your attention, your willingness, and your nice lecture today.
Thank you very much. I apologize for the technical glitches.
No problem. That's what matters.
Bye-bye.
Okay, one more, Professor Hanry Yu. Can we all take some pictures together?
Yeah.
So all of us, please open your video first, because the secretary of doctoral program will make a picture of all of us. Who will give? Nova or Unita, can you give the 1, 2, 3, 1, 2, 3?
Okay, Dr. Radiana. One, 2, 3. Once again.
Yeah.
One, 2, 3.
Okay. Once more? Is enough?
I think it's enough.
Okay.
Enough, Prof.
Okay, thank you so much. So, Prof. Hanry Yu, we'll see you at the 1st of December, okay?
You have another lecture, for the next December.
Yes.
Thank you very
much. Thank you, Prof. Hanry Yu. See you.
See you.
All right. Thank you so much.